Okay, thank you. Introduction. Hello, everyone. My name is Yuji Sukekawa from the University of Tokyo, Japan. Today, my presentation title is Evaluation of Background Odor Influence on Mold Odor Detection using Multiple Insect Olfactory Receptor-Based Sensor Arrays. Okay, this is outline of my presentation. First, we will explain the motivation of this study and our insect odor sensing technology. Next, we will explain a method for odor discrimination based on the sensor array, and then show the result of evaluating the response to various odor components and evaluating the influence of background odor components on odor discrimination. And finally, we will summarize conclusions and present future prospects. Okay, firstly, I would like to give you some research background and context. The contamination of agricultural crops is one of the most important problems affecting food safety. This diagram shows the flow of agricultural products from production to consumption. In this process, various contamination risks exist. One of the contamination risks in the production stage is mold contamination of agricultural crops. This picture shows the fungal disease of wheat called Fusarium head blight, FHB. FHB is caused by mold fungus, Fusarium graminearum. This fungus is known to produce mycotoxins such as deoxynivalenol and nivalenol. So it is necessary to prevent contamination and detect it in its early stage. So how can we detect mold contamination? Our solution is using odorants. Mold emits several volatile organic compounds, VOCs, such as 1-octane-3-ol, 3-octanol, and 3-octanone. These exist as a mixture with some other compounds. If we can detect these kinds of mold-characterized VOCs, it may lead to finding early-stage fungal disease, and also it may lead to preventing a spread of mold contamination. The next question is, how can we detect these moldy VOCs? Our answer is insect. A wide variety of insect species inhabit the earth. It is said that there are more than million species of insects in the world. Insects can sense a lot of odorants through olfaction. For example, fruit fly. Maybe you have seen at the sink in your kitchen. They can detect moldy odorants in order to avoid mold-contaminated food. Likewise, bedbugs and other insects respond to several odorants and pheromones. So we can say that insects are professionals at odor detection. To utilize insect olfaction, our group has developed sensor cell technology. The left picture is an image of sensor cells. The cell has ion channel type insect olfactory receptor and calcium-sensitive fluorescent protein, which were genetically introduced. So when the odorant binding to the receptor, the ion channel opens and cut ion, including calcium ions, influx happens. The calcium ion binds to the fluorescent protein. Finally, the fluorescent intensity change happens. So this cell works as an odorant-sensitive indicator. The right figure shows the example of fluorescence. This figure shows the sensor cell response with the food color overlay. You can see the increase of fluorescence intensity when the cell responds to an odorant. One of the advantages of the sensor cell is that the response profile can be modified by changing the expressing receptor. For example, regarding the receptor expression in sensor cells, Drosophila fruit fly has 60 OR genes, and an adult fly uses 35 ORs. According to the public database, some of OR respond to mold VOCs such as 1-octane-3-ol and so on. We have developed 4 sensor cells with or 13A, or 47A, or 56A, and or 98A receptor from Drosophila. So in this study, our research objectives was to construct a sensor array using these 4 sensor cells and clarify the response profiles to various odorants, as well as evaluate the capability to discriminate odor components. Furthermore, we also aim to evaluate the influence of background odor components on the discrimination of multi-odor mixtures using artificially prepared samples. For the response profile evaluation, the sensor cell we used, for the response profile evaluation of the sensor cell, we used fluorescent microplate reader system. By using this system, we could quickly evaluate the cell's response to the odorants. Okay, in this slide, I will explain the method of constructing the sensor array using multiple types of OR sensor cells. From each sensor cell, we can measure the fluorescence response as shown in this illustration. To convert this time-freeze data into a scalar value, the area FA between response curve for order A and negative control curve was calculated. Then FA value was normalized by using positive control value, and this normalized value RA for each OR sensor cell is combined as multidimensional data. That is the output of virtually constructed sensor array. Moreover, to visualize the distribution of response pattern of this multidimensional data, principal component analysis, PCA, was used. Also, for classification of tested odorants, support vector machine, SVM, classifier was used and evaluated the classification ability. Okay, from this slide, I will show some experimental results. This figure shows the selectivity of 4 sensor cells. We tested 10 different odorants as shown in the right side of the slide. We can see the different response profile. Each sensor cell has a different molecular selectivity. In particular, focusing on 3 multi-odorant components, they show obvious difference. To visualize this difference, we applied PCA. This figure shows the result of PCA. Although there are some overlapping points, you can see that many of the samples are distributed individually. When I overlay the structural formula onto the PCA score plot, you can find that the odorants with similar structure are distributed relatively close. For example, a difference between these two is existence of double bonding, and also these two difference of carbon chain lengths. Therefore, this sensor array could be useful for ordered components discrimination because of its ability to capture molecular features. In addition, we tried to build a classification model for ordered components using the same data set. Okay, because my presentation time is limited, I will focus on the important points. We used a support vector machine classifier to train. We used a support vector machine classifier and trained a model by using 80% of the sample points from the dataset as I mentioned earlier. Through 5-fold cross-validation, hyperparameter searching, and model training, the classification accuracy became around 98%. Furthermore, we tested classification model on the remaining 20% of dataset. It was able to successfully classify all of the test dataset. The reason for this is that sensor array, which uses multiple insect OR, is able to capture the characteristics of each odorant molecule so that dataset can be successfully classified using the simple classification model. Moreover, the response pattern of the target multi-odorant mixtures in the presence of background odorant components were evaluated. Here, we prepared single and mixture of 3 target MODI odorants. We then checked how the response pattern changed depending on whether or not heptanol was included as a background odor component. The figure on the right side shows the measured response pattern. The right half of the figure contains the background odorant. From this result, you can see the relative response profiles preserved, even though the background odor affects the response value. Here, we applied SBM classification similarly to previous slide. We trained the classification model so that it could classify the target odor components into 8 different categories, regardless of whether or not there were background odor components. I will not go into the detail of the classification accuracy of the model by cross-validation was around 81%, and the classification accuracy for the test dataset was around 84%. So it was confirmed that the classification was relatively successful. On the other hand, if you look at the sample that failed to classify, you can see the you can see from the figure that it was all 3 combinations and 2 combinations of 1-octantriol and 3-octanol. The reason for this may be that the sensor response is near saturation, and the difference between samples are not being measured well. Actual sample will have a combination of more components, and it is necessary to select sensor elements that do not respond to these components or that do not saturate the sensor response, even if they do respond. Nevertheless, our sensor array may allow the possibility of discriminating between odor mixtures even in the presence of background odors. Okay, this is the summary of my today's talk. This study is an important step in the development of an odor biosensor to detect mold contamination of food and agricultural products. And then our next work is evaluation of odor mixtures with increased number of the background odor components. And also, for practical application, real mold contaminated sample should be used for sensor evaluation. Okay, this is the final slide. Thank you for your kind attention.